Yeah, I think we'll get started.
Well, maybe I just say a few words and maybe Ezra can say a few words too. So welcome to this public thesis defense of Joe. It's after a very long journey, but now he's got the last hurdle, which is to explain it to the public, what he's been doing. And so we look forward to the presentation. This thesis has got several supervisors, including Ezra. Ezra, do you want to say a few welcoming words?
Just that I've had the pleasure to know Joe already before the thesis, when we started off with the compound action potential that we have been coming back to at the end of the thesis again, and it has always been very enjoyable to work with Joe and to bounce ideas back and forth and to benefit from his rich backgrounds. And I think Joe knows that as long as he wants, he will always find open doors with our laboratory. It shall always be my pleasure to collaborate with him.
Wonderful.
Thanks.
Michael has also been very close. Do you want to say a few welcoming words?
Yeah, it's been amazing working with Joe all this time. He's been really a very talented and astonishing PhD student. It was always, when you're busy and you give him something to do, you think it will take him a week, and the very same day he comes back with the results. It was always really impressive what he managed to accomplish.
Wonderful. Joe, the floor is yours. We look forward to your presentation.
All right, thanks everyone. And thank you to everyone who came, both in person and on Zoom. So, the title of my thesis is Coupled Electromagnetic Electrophysiological Modeling of Neural Circuits. So this leads to an obvious question, which is, Joe, why doesn't your projector not work? There we go. The obvious question is, what are we talking about here? So the basic idea behind this thesis is that we want to understand what the electrical signals that we record in or on the brain tell us about the underlying neural activity. And there are two broad classes of recording modalities that we're interested in. The first is the EEG. You might be familiar with this. This is when you place electrodes on the surface of the skin. It's a very commonly used diagnostic tool, particularly for epilepsy and sleep disorders. And it's also used in basically every single field of cognitive science. The other modality that we're interested in is the local field potential, or LFP. And this is when you stick electrodes inside the brain. As you might imagine, it's pretty hard to get people to volunteer for experimental brain surgery, so this is mostly limited to animal research. However, there are some emerging clinical uses of LFP, particularly in the context of deep brain stimulation. So, we want to know what these signals tell us about what the brain is doing. But in order to answer that question, we first need to answer a different question, which is, where do these signals physically come from? And at a fundamental level, they come from neurons. Now, what are neurons? Obviously, neurons are just brain cells. And like any cell, they have there we go. Like any cell, they have a cell body. Inside that cell body is a nucleus and a bunch of other organelles. But I'm an engineer, not a biologist, so we're going to pretend that doesn't matter. Instead, what I'm interested in are the axons and the dendrites. These are the projections from the neuron which connect to other cells. And it's through these connections that communication happens. And it's this communication that results in information processing and everything else that makes the brain work. So when I ask the question, what does EEG or LFP tell us about the brain? What I'm really interested in is, what do EEG and LFP tell us about how neural connectivity and neural communication is happening? So if we zoom in on the neuron, we can see that it has a cell membrane like any other cell. This is made up of a phospholipid bilayer, which creates the structure of the membrane itself, and a bunch of channel proteins inserted into the membrane. But again, this is starting to look awfully biological, so we're going to want to simplify this. And the advantage of simplification is that we can now abstract away a lot of the complexity and implement these as models, and those models can tell us a lot about what the brain is doing. So the way we do that is as follows. We start off with a neuronal morphology, that is just the shape of the neuron. We're not interested in what goes on inside. We then discretize that morphology into cylinders, which we refer to as neural segments or compartments. Then, we can treat each of those compartments as an RC circuit. That is a resistor and a capacitor. This capacitor represents the membrane itself, and this resistor represents the channel proteins, which allow for the flow of ions from the inside to the outside of the cell. The inside of the cell here is modeled by this series of resistors, which connect each segment to its neighbors. And the outside of the cell is ground, that is, it has zero connection. So, if we have a current inside the neuron that arises, for example, from an electrode or from a synapse, that is, contact with a neighboring cell, the current flows across these resistors and across the capacitor outside of the cell. And by Ohm's law, the flow of the current across the resistor is given by I equals V over R, where I is the current, V is the membrane potential, and R is the resistance. And similarly, the flow across the capacitor is given by the equation I equals C dV dt, where C is the capacitance of the membrane, which is just a physical quantity of the membrane itself. And this V, again, is the membrane potential. So this leads to an easy series of differential equations, which we can implement, in our case, in the neuron solver, and that tells us the behavior of the neuron. But for those of you who have some engineering background now, this is just a series of RC circuits, and that just acts as a low-pass filter, and that's not particularly interesting. So what we need to understand is how this leads to communication, more specifically, how it leads to the propagation of signals. So we can see how that works if we zoom in on one of these segments, because what I'm showing in this slide is a simplification. In reality, we modeled each of these segments in a bit of a more complex way. Yes. So instead of one resistor, we have several, each of which represents a different channel protein, and each of those channel proteins allows for the passage of a different type of ion. And the interesting thing is that these resistors are actually dependent on both time and the membrane potential itself. So here's what happens. Suppose you have a current entering this particular segment. That's going to depolarize the membrane, meaning that the voltage increases. When that happens, the resistance of the sodium channel decreases, allowing for the flow of sodium from the outside of the cell to the inside of the cell. This further depolarizes the membrane potential, causing the resistance of the sodium channel to decrease more, and so on, leading to this, what we call an action potential. At the peak of the action potential, the potassium channel is activated. This allows for the flow of potassium from the inside of the cell to the outside of the cell. And this results in the membrane potential dropping back to its initial value. And as you might imagine, with all these flows of currents, some of that current is going to flow from this compartment into its neighbor, allowing for the propagation of the signal. And that, fundamentally, is how a neuron works. But, of course, that's just one neuron. And the work in this thesis relies on around 4.2 million. Specifically, it relies on the Blue Brain Project's model of the rat somatosensory, non-barrel somatosensory cortex. It has 6 layers, each of which represents a different set of cell types. And all of these cells are connected according to biological rules. You'll notice that there is this one column of around 210,000 neurons that's sticking out. That's just for visualization, don't worry. But this is the O1 circuit, which we use in a lot of cases to reduce the computational complexity of this entire simulation. But I still haven't answered the initial question, which is how all these simulations produce EEG and LFP signals. But the answer is actually basic physics. For the range of frequencies that we're interested in, the signal that's produced by any transmembrane current is directly proportional to the current itself. That's because tissues are ohmic at these frequency ranges. So what that means is that in general, the signal that we record from these simulations is just a linear combination of all of the transmembrane currents in all of the compartments in our model. So the key then is that we need to find these coefficients, C sub j, for each of the transmembrane currents. How do we do that? Well, the easiest way is to do something called the point source approximation. There we go. The point source approximation. Here, what we do is we assume that the brane is an infinite and homogeneous space. At that point, the coefficient C sub j is just given by the distance between the electrode, R sub e, and the neuronal segment, R sub j, and the conductivity of the tissue, sigma e. Now, the assumption that the brane is infinite and homogeneous is actually a pretty good one when we have an electrode inside the brain surrounded by a relatively large volume of bulk tissue. However, it's obviously not true if you've got an electrode on the surface of the scalp like you do for EEG. In that case, we decided to use something called a reciprocity-based approach. So the idea here is that you have a model of your brain, or your skull, and you have two electrodes on the surface at points A and B. Those are your recording electrodes, your EEG electrodes. So, assume that you run a current between these two recording electrodes. That's going to result in an electric potential inside the volume of tissue. Then, you can interpolate this potential at the locations of the neural segments. Here that's R1 and R2. Then, you multiply those potentials by the neuronal currents, I1 and I2. There we go. And then you sum over all of the compartments. And that gives you your coefficients, or that gives you your EEG signal. And then, obviously, the coefficient itself is just the product of the potential at the location of the compartment, divided by the current that you apply. The advantage of this approach is that we have to make far fewer assumptions. In particular, all we need to assume is that the tissue is ohmic and that the neural segments can be approximated as point sources. However, this isn't what most people in the field actually do. Instead, they go a step further and use a dipole approximation. They make the assumption that the E field, that is, the gradient of the potential field phi, is constant over the scale of the neural source. In that case, you can take the Taylor series approximation of this equation, and then your signal is given by the E field at the location of the neural dipole, multiplied by the dipole itself, which is the vector between your neural currents, or I1 and I2. And I'll explain in a little bit why we chose our more general approach over this dipole-based approach. But of course, the next question is how we actually calculate these potentials. And it's a hard problem, so we divide it into thousands and thousands of little problems using something called the finite element method. We take a volume of tissue, and we divide each of the tissues within that volume into tetrahedra. And then our finite element solver is able to impose boundary conditions, which give you the particular quantity that you're interested in our case, the electric potential. And so we created a finite element model of the rat head that's aligned to the BBP model of the somatosensory cortex. And this model contains all of the tissues that have a significant impact on the signal. Here I've just highlighted a few: the skin, muscle, skull, and of course the brain. So now that I've shown you all these methods, the next question is, how do we implement them? The naive approach, which is what I did at the start of my PhD, would just be to run this simulation, save all the transmembrane curves, and then apply one of these calculation steps as a post-processing step. The problem with this is that it results in storing hundreds of terabytes of data, and it crashes the file system every time that you try to run it. So they forced me to come up with something else. And that something else is the blue recording system, a pipeline for the efficient calculation of extracellular recordings in large-scale neural circuit models. I won't get into too much detail, but the basic idea of blue recording is that you pre-calculate these coefficients, C sub j, write them to an HDF5 file, and then at runtime, the neurodamus simulator will read the file and calculate the potential resulting from all of the transmembrane currents at each time step. There are, of course, other software packages that do something similar, but compared to those, Blue Recording is, first of all, 3 to 4 times faster than its nearest competitors. It's also compatible with the Sonata format for circuit specifications, meaning it's highly flexible. And most importantly, it implements the general form of the reciprocity calculation, which none of the competing packages do. Now, why is that important? So what we did to show why it's important is we calculated EEG, ECOG, and LFP from the entire somatosensory cortex, using all 3 of our calculation methods. And what we can see is that clearly, the general reciprocity approach and the dipole-based approach diverge. Now, given that the dipole-based approach is just a simplification of the general reciprocity approach, the implication must be that one of the assumptions behind the dipole approach isn't met. And in particular, what we're able to show is that the assumption that the E field is constant over the cortex does not hold, especially in the case of ECOG electrodes. Here we see the ECOG electrode in the somatosensory cortex model. And in this background, you see the E field that's produced when you run a current between the recording and the reference electrode. And obviously, over the scale of the somatosensory cortex, the E field varies significantly. Now, what this means is that, basically, in order to get accurate values for ECOG and EEG calculations from simulations like this, you have to use the general reciprocity approach. So, the Blue Recording package is available on GitHub under an open source license, and I encourage any of you who are interested to check it out. So, now that we can do all this, what can we learn? In particular, one question that we're interested in answering is, what's the biological basis of the whisker flick EEG? Now, this might seem like an oddly specific question, but it's an interesting one. First, because whisker flick is a very commonly used model of somatosensory processing. So what we learn about whisker flick in rodents can easily be translated to other species and other modalities. It's also a fairly convenient question to answer, because the somatosensory cortex model is optimized in order to replicate firing rate responses from whisker flick stimulus. So this is the somatosensory evoked potential. It's the EEG that's recorded in response to a whisker flick. And as you can see, over the course of development, it changes significantly. Starting from P10, that's postnatal day 10 days after birth, to P21, 21 days after birth. The shape and the amplitude of the SEP change a lot. Now, the SEP has a couple of canonical features. The first is the P1 component. That's this initial positive deflection, which arises around P16. The second is the N1 component, this negative deflection, which is present from at least P10, but gradually narrows as the animal ages. So in order to model whisker flick, we use 2 different versions of our somatosensory cortex circuit. In both cases, we're using our 7-column subvolume with 210,000 neurons. In the first case, we refer to it as the original circuit. In the second case, we refer to the so-called Schneider-Nizzel connector, or SM circuit. In this circuit, we've rewired some of the connectivity to better approximate data that was obtained from electron microscopy. In particular, we moved synapses, that is, connections between neurons, between basket cells and pyramidal cells, which are these large neurons that span the width of the height of the column, from the apical dendrites up here down to the basal dendrites in the perisynaptic region here. So when we simulate these circuits, what we see is that, first of all, the in silico models produce a reasonably accurate EEG. Here we've scaled the P1 component amplitude of the in vivo circuit to match in silico, but we can see that the width of the P1 component is approximately the same in both in vivo and in silico. The amplitude of the N1 component is also correct for the original circuit, and then we can see that for the Schneider-Nizzel circuit, it's significantly bigger, and I'll explain why that is later. We also see, obviously, that the M1 component is around 3 times too wide in our models compared to in vivo. But we have a reasonable explanation for that. If you're interested, you can ask at the end. But the question that we really want to answer is what this tells us about neural connectivity. So we're able to break down the EEG signal into its constituent components. That is, what connections between the presynaptic cells and these pyramidal cells lead to the EEG. And what we see is that for the P1 component, the signal is primarily driven by thalamic input, that is, input from lower brain regions to the cortex. However, interestingly, there is also some modulatory effect from layer 5 inhibitory cells. That means that both of these features could explain the changes that we see over the course of development. Now, as for the N1 component, things are, of course, much more complex. Again, there is a strong contribution from thalamic input, but we also see large contributions from both cortical-cortical inhibition and excitation. And in the Schneider and Zal connectome, we see a fairly similar result. However, we see a much larger contribution to the N1 component from inhibitory activity. So why is this? If we break down the sources of the N1 component further, we can see that it's primarily due to these parasomatic targeting cells that account for almost the entirety of this difference between the original and the Schneider and Zal connectome. If we look at the firing rates of the parasomatic targeting cells in the tube models, we see no significant differences. So in fact, it is the location of the synapses that has the major effect. In particular, when we move synapses from the apical dendrites to the basal dendrites, we're moving them from a region that has more positive compartment weights to a region that has more negative compartment weights. That means that if you have an inhibitory current arriving in the parasomatic region and a corresponding excitatory current returning outside from the apical dendrites, you'll get a much larger negative deflection than you would if you had uniformly distributed synapses. In particular, you could say that the neuron acts in a more dipolar way. So the key takeaways are, first of all, that the P1 component is driven primarily by thalamic input, but modulated by layer 5 inhibition, and that the N1 component is driven by a mix of thalamic and recurrent sources. Most interestingly, we've shown that rewiring the connectome can have significant impacts on EEG without changing firing rates. There's a dissociation between these two metrics. And what this means is that we could actually use our EEG signals recorded from these models as an additional way to constrain the development of our models themselves. So I talked about EEG. The next obvious step would be to look at the elephant from this report. But we tried to do that, and we got some very interesting results, which I'll talk about now. So one thing we're interested in is solving the inverse problem. If in LFT, you stick an array of electrodes into the brain, and you record voltages from all of those electrodes. So what we want to know is what's the distribution of currents that produces that set of voltage shifts. And this is a hard problem to answer, but if we make some assumptions, it can be more tractable. In particular, we assume that currents are homogeneous in the plane perpendicular to the electrode. And we assume that they're homogeneous around a certain radius around the electrode and negligible elsewhere. So under that assumption, we can calculate something that we call the inverse current source density, which is a very commonly used metric for CSD. But what's happened recently is that we've developed, not we, but the community, has developed new generations of LFP electrodes, such as neurolasers, which have resolutions far higher than anything that's been seen before, going down to almost 15 microns between electrode contacts. And people have been using these electrodes in vivo, and they found some surprising results. In particular, they've seen that at high density electrode arrays, there are high frequency oscillations between current sinks and sources in the CSD that don't occur in less dense electrode arrays. And there are two possible reasons for this. And it could be that, in fact, we are now resolving new features of the CSD that we weren't able to before. But it could also be that because CSD makes certain assumptions, and those assumptions don't necessarily hold when we're using these very fine electrogalleries, that we're producing spurious results. And I believe that our models can help us to understand that better. So here we do see that we get the same sort of thing in our models. We calculate LFP from an electrode array ranging in width between 160 microns and 20 microns. And we can see that starting at around 40 microns, we get these high frequency oscillations between sinks and sources. So in order to determine whether these are real or not, we developed a metric called objective CSD. Here what we did is we took our electrode array and we defined a disk with a certain radius rho around it. Then, for each neural segment that's within the disk, we assign a coefficient of 1. And for each neural segment outside the disk, we assign a coefficient of 0. Then we just sum all of the neural currents scaled by the coefficients. Basically, that's the same as taking the average neural current over the disk. And what we see is that objective CSD diverges from the ICSD metric, which is used in clinic. In particular, starting at around 40 microns, the correlation between the two metrics begins to drop. And as I mentioned before, that's also the density at which we start to see these high frequency oscillations between sinks and sources. But this doesn't tell us yet whether these sinks and sources are real or whether they're artifacts. So if they are real, why isn't it that we see them in the objective CSD? Well, it could be that because we're averaging over a very large radius, we average out high frequency oscillations that occur close to the electron. Thus, if we reduce the radius for our OCSD metric, then we should begin to see these oscillations emerge. And consequently, we should see an increased correlation between ICSD and OCSD. Alternatively, if they are artifacts, then we can develop another metric, which we call non-negative CSD, which can identify them. Basically, the idea is that we take the second derivative of the coefficients for the LFP. That gives us the coefficients for CSD. Then we just zero out all of the coefficients which have negative values. And that should, in principle, eliminate any artifacts. So if these oscillations are actually artifacts, then what we expect is that we see a higher correlation between between OCSD and non-negative CSD than we do between ICSD and OCSD. So that's exactly what we did. And we see a couple of interesting things. First of all, we see that at 50 microns and 100 microns, there is a stronger correlation between ICSD and OCSD than there are at other radii. But once we drop down to a 20 micron radius, the correlation drops further, which, if this were actually true oscillations that we were seeing, we should see the correlation increase even at 20 microns. We also see that the correlation between OCSD and NMCSD is consistently higher than the correlation between ICSD and OCSD, which again is further evidence that these are actually artifacts. Instead, what we think is happening is that when we go down to 50 or 100 micron radius, we're best approximating the radius within our circuit model, where the currents are actually homogeneous. And there we expect to see the best results for ICSD. So in order to determine if this is the case, we then look at the correlation between the so-called standard CSD metric and OCSD. Standard CSD doesn't consider any radius over which the currents are assumed to be homogeneous. So we would expect to see any increase in the correlation between OCSD and standard CSD with reduction in rho to be associated with resolution of true oscillations. But what we actually see is that there is no improvement in the correlation between OCSD and standard CSD with rho. Instead, the correlation between between OCSD and non-negative CSD is consistently higher. So what this suggests is that for dense electrode arrays, the CSD metrics that are commonly used in experimental labs might actually produce spurious results. What this means is that CSD estimation methods might need to be optimized for particular experimental conditions, for particular widths of true current distributions. And as we showed in our paper, which is going to be coming out soon, but which I don't have time to discuss in this presentation, in silico simulation can actually help to come up with optimal parameters. So now we'll move on from the brain into the peripheral nervous system. In particular, we're interested in vagus nerve stimulation. So vagus nerve stimulation is a technique that's used for a wide variety of conditions, but in our case, we're interested in control of cardiac function. Our experimental collaborators in Pisa implanted a so-called V-ray electrode, that is an electrode that's inserted across the nerve into the right above the cardiac branch of the vagus nerve in mini veins. They also insert a recording electrode around 6 centimeters away. Now, you can see that the vagus nerve has this very complex multifascicular structure where each of these fascicles is a bundle of nerve fibers that's surrounded by a highly insulated tissue. So what we want to do is simulate the simultaneous stimulation and recording of the vagus nerve. The problem is that there are around 300,000 fibers within the vagus nerve, which is too many to model explicitly. So instead, we developed a reciprocity-based semi-analytic approach. I won't get into the details now, but the basic idea is that we obtain single fiber action potentials using the reciprocity theorem. We then assume a set of uniform fiber diameters within the nerve. Then, for each diameter bin, we scale the single fiber action potential by the fiber density and by the probability of fiber recruitment, which also depends on fiber diameter. Low diameter fibers are more difficult to recruit with electrical stimulation than high diameter fibers. So what we see is that our model is able to replicate the in vivo compound action potential signal. In particular, we get a reasonably good estimate of both the amplitude and the shape of the signal. And we also see that as current increases, the shape of the signal saturates, which is what we also see in vivo. Now, if we break down this signal by the contribution from each of the fascicles, we see something very interesting. The contributions from each fascicle tend to cluster. We see one cluster in the middle that's associated with the fascicles in which almost all of the fibers are activated. You can see that here. There's a very sharp threshold in the fiber diameters that are activated, around 1 micron. And you can see that these fascicles produce these very smooth compound action potential curves. However, we also see two other clusters associated with fascicles further away from the electrodes, here in green and orange. Here you get a different sort of transition between the fiber diameters that are activated and the fiber diameters that aren't activated. And these fascicles tend to produce these very irregular compound action potential contributions with large deflections at around 4 milliseconds after the stimulus. So the reason for this is actually because, in most cases, the single fiber action potentials tend to cancel out. If you activate almost all of the fibers, then you get this very smooth distribution of single fiber action potentials, where the peaks of one diameter correspond with the troughs of the neighboring diameter. And that means that you get this very smooth signal where most of the amplitude of the signal is canceled. However, if you have a sharp transition between the fiber diameters that are activated and the fiber diameters that aren't activated, the smallest diameter fibers, which are activated, don't have any smaller diameter fibers to cancel out their contributions. And thus, you get this very large deflection towards the end of the signal when the signals from the smallest fibers arrive at the recording electrode. So this actually has some very interesting clinical implications. In particular, the assumption that's commonly made in clinic that the amplitude of the evoked compound action potential correlates with the amount of activation is simply not true. In fact, what we see in our model is that as you increase the stimulus current, you first start to see an increasing amplitude from the compound action potential due to the activation of more and more fibers. However, after a certain point, when you activate more fibers, they begin to cancel out, reducing the amplitude of the compound action potential. And what this means is that the overall stimulus versus amplitude curve is non-monotonic. And this means that our assumptions about how we actually calibrate vagus nerve stimulation in experiments or in clinic need to be reassessed. So to conclude, we've shown that semi-analytic modeling can accurately replicate in vivo ECAPs. We've shown that incomplete activation of the population of fiber diameters results in significant impacts on the shape and the amplitude of the ECAP. And we've shown that ECAP amplitude is certainly not a perfect measure of fiber activation. And what this suggests is that semi-analytic models can actually be used to optimize our recording setups to maximize information content, which may have important implications in the future for closed-loop stimulation of the vagus nerve. So, to summarize the last 6 years of my life, we've developed software to efficiently calculate extracellular recordings in large-scale neural circuit models. We've used this software to characterize the biological origins of the whisker-flecky gene. We've shown that the current source density metrics that are commonly used can produce spurious results for dense electrode patterns. And we've shown that evoked compound action potentials in vagus nerve stimulation are also much more complicated than previously believed. So to summarize, my thesis has caused a lot of problems for experimentalists, and maybe in the future I'll help to solve them. So with that, I have a lot of people to thank. I won't get to you all by name, so forgive me if I don't. But I have to start off, of course, with my supervisors, Henry Markham, without whom none of this would be possible. The Blue Brains come to a close, and I think it's very clear that we've accomplished so much together. So Henry, you owe yourself a round of applause. I also have to thank my section manager, Michael Ryman. You've been an inspiration to me, both personally and professionally. I think our work together has been really productive, and I look forward to continuing it when we wrap up these papers. I owe a huge debt of gratitude to Ezra Neufeld, my co-advisor. I was a master's student whose project has just fallen through when Ezra took a chance on me and invited me to Zurich. That led to possibly the most important scientific collaboration of my life. I'm so grateful to you, Ezra, for all of your effort, for all of your support, for all of your care, and I'm very thankful to be able to work with you after this. Finally, I need to thank Felix Sherman, my master's thesis advisor, who was absolutely invaluable in the development of the software blue recording. He was not only a mentor to me professionally, but he also taught me how to actually write good commented code, and he would always remind me to remember to label my videos. So Felix, thank you very much. Then I need to thank the Connectomics team at the Blue Brain Project. Christoph, Natalie, Daniela, Michael, of course, Andras, Steve, James, and Sirio, who is not in this picture. You guys have been so great, professionally and personally. You're the reason that I got into bouldering, so thank you for giving me a hobby. And yeah, without you, this would not have been possible. I also need to thank a lot of people from the HPC team, Jim, Pramod, and Sergio for forgiving me whenever I crashed GPFS, and for helping me to make my code a little bit less brute force. And most of all, Jorge, who was second author on the blue recording paper, really should have been co-first author. Without him, none of my work would have been possible. Then I also need to thank a couple of people at the EDIS Foundation, Antonino, who basically taught me how to use Sim4Life, Taylor, who taught me how to use the entire Blue Brain Project software stack, and Christina for all of her work in helping me with presentations like this one. And then there's everyone at the Blue Brain Project who I didn't mention by name, but who have been absolutely invaluable in everything that I've done, from writing my papers to booking travel. Thank you all. Then there are the people whose impact on me has been more personal. I'll go in chronological order, starting with my family. I joke all the time that my mom's a psychologist, my dad's an electrical engineer. So I was born and bred to be a computational neuroscientist. You think that's a joke, Jake is also a neuroscientist. But seriously, Dad, if it wasn't for your early introduction to math, to analytical rigor, to fascination with technology, I wouldn't have done this. And Mom, if it wasn't for your lifetime of helping to fix people's brains, I just would not be here today. Few more early influences on my life. My high school math teacher, Mrs. Petsu, she introduced me to the idea of mathematical modeling when I was 13 years old. She also introduced me to the idea of actually working on my homework, so thank you. And of course, Uncle Peterchev, my bachelor's thesis advisor, we worked together for 4 years, and he is the first person who taught me how to be a scientist. Thanks. Then, of course, there's my other family, my friends from Duke. We've been together for 10 years now, longer outside of college than we knew each other in college. We've been together for each other's thesis defenses and our weddings, and potentially soon our children's births. You guys are also my family. Thank you so much. I would not have gotten through this PhD without our monthly Zoom calls, our weekly chats, or daily text messages. Then last, but certainly not least, my best friend and partner, Swati Sharma. Swati, without your constant love and support for the last 2 years, I would not be the man that I am today. Thank you so much. You are so important to me, and I'm so glad to have you in my life. You deserve your name on this thesis too. All right, so with that, we can move on to the
Thank you.
ceremony.
Just one little formality. Before signing, there has to be at least 2 questions. They have to be from the public, at least 2 questions.
Can you explain why the N1 component is longer?
Sure. So we've got two potential hypotheses. Yes. So the first hypothesis is that we're actually missing half of the brain. People have shown previously that the drive from the contralateral cortex actually has a significant impact on the EEG reported in the ipsilateral cortex, starting at around 26 milliseconds after the stimulus, which is exactly when we expect the N1 component to recover. And in fact, it's also been shown that around it's been shown that it's around P16, which is when the N1 component starts to narrow, is when the contralateral cortex starts to be affected by neural activity from whisker flight. And as we've seen, the N1 component in our model is consistent with the width in around postnatal day 10, when there is no activity in the contralateral cortex. So that could very well explain the width of the N1. The other hypothesis is that it has to do with the distribution of thalamic inputs onto layer 6. People have shown before that thalamic inputs tend to preferentially target basal synapses, rather than apical synapses. And basal synapses are associated with shorter EPSPs than synapses onto apical dendrites. So if we actually implement that dendrite preference in our thalamic innervation, that could also shorten it.
It can also be a very general question, like, how is it going to help my work in psychology, or what kind of
Yes,
new
ma'am.
engineering?
Can you tell us more about the problems that you've created for the experimentalists like your brother, and how maybe your work will eventually provide solutions as well?
Sure. So, like I've shown, the CST metric can produce these strange high-frequency oscillations when you have a very dense electrode array. Now, specifically, the reason for that is because the contribution from a current source located on the, assuming that it's centered on the electrode array, is a function of both the distance between the electrode contacts and the true width of the current source. So there are certain values where you get these negative contributions and values where you get positive contributions. So what my work can actually do is to estimate what the true distribution of the current sources or of the width of the current sources in the cortex actually is. And we can do that by using the objective CST metric. So for experimentalists, what might be interesting is to develop models of their particular experimental system and their particular electrode array, and then estimate the width of the true current source distribution, and then find where they are along this fit diagram, such that they're using a electrode density which minimizes the range of negative values while also maximizing array density.
Any further questions? You all agree you can get a PhD? Kind of. That's right. Come and sign. Okay. Wonderful. Congratulations, Joe. It is official. Wonderful.
Thank you.
Now is the next step. What happens after the PhD?
So I'll be spending the next 6 months working with Ezra, finishing up my papers, and focusing on some of the cool applications of this vagus nerve model. So I'm very much looking forward to it.
Opening new questions.
Good luck, Ezra. Wish you all the best.
Thanks.
Joe. And Ezra.